Hello everyone. This video will discuss population ecology. Um, and to introduce a couple core concepts that will be discussed in the video, what we will talk about will revolve around these four main topics. The first one, a population consists of all of the individuals of a given species that live and reproduce in a particular place. Second, population size can increase or decrease over time. I think that should be obvious. Three, the age structure of a population helps ecologists understand past changes and predict future changes in population size. And the last core concept is that the dynamics of populations are influenced by the colonization and extinction of smaller interconnected populations that make up what we know of as a metapopulation. So we'll be discussing those four major concepts throughout the video. I want to start with an example to sort of set the scene and introduce you to the idea of population ecology. You'll see an image of a butterfly, and in 2004, a group of students who were on a field trip in the Dominican Republic, an island that's found in the area called Hispaniola, swept up an unfamiliar black and white butterfly, and that butterfly that they found is called a lime swallowtail. And it was the first of its species to be found in the Dominican Republic. It's typically native to Asia, but it was the first time it was captured in the New World. And in basically no time, this lime swallowtail spread to all of the corners of Hispaniola, which was a pretty serious concern because the caterpillars that will eventually become this butterfly that you see strip the leaves off of young lime and orange trees. And within 3 years, the lime swallowtail had established itself in the nearby island of Puerto Rico and Jamaica, and it has now reached Cuba as well. And from Cuba, it's just a short flight to Florida, and the multi-billion-dollar citrus industry that we have in the United States could essentially be wiped out by this invasive species. So the question I have for all of you is one that we will try to answer by the end of the video. And it is, can we enlist basic principles of biology to help understand this butterfly's Caribbean expansion and to predict its future as it continues to spread throughout the Americas? So in order to answer that question, those two questions, we have to start with some basics. So the first thing I would like to define is ecology. What is ecology? Ecology is the study of the relationships of organisms to one another and to the environment. And when we think about ecology, we should also think about evolution because ecology and evolution are very closely related. It's through natural selection that populations can adapt to the abiotic and biotic components of their environment. And we would say that abiotic factors are the physical factors that influence evolution, and those could be the climate or possibly nutrient availability. Whereas the biotic factors or the biological factors would include competitors, predators, parasites, prey, as well as organisms that provide food or shelter. And through it is the traits that organisms have that have evolved under natural selection that determine whether a population will grow in size or shrink in size under a given set of physical and biological conditions, perhaps disappearing entirely from a local environment. And you'll see that there are four images on the right side of the screen. And as we move down the screen, we increase in terms of what each of those terms include. So let's start at the top. Individuals are just individual organisms, so one organism that we're talking about. And individual organisms make up populations, which are groups of organisms that can interbreed with each other. And populations make up communities. and communities could be defined as populations of different species that interact with each other within a locale, which would be a location or some space. And then many communities together would make up an ecosystem, which could be defined as all the living organisms as well as the non-living elements that interact in a particular area. So all of the biotic and abiotic factors that are in some space, that would be what we would define as an ecosystem. So this video that we will watch right now talks about population sizes and human population growth over many years. So let's watch that now. So that brings us to the question of how populations are characterized. The video really talked about size and how populations are growing in size. But populations can be characterized not only by size, but also by range and density. And so those three key features of populations are how we define um, the healthiness of a population and the ability for it to go extinct or possibly take over a certain ecosystem. And we can use mathematical and analytical tools in order to be able to predict those changes depending on population size, range, and density. And then we can use those mathematical and analytical tools, those predictions, to be able to better design management strategies to help us save threatened species, or be able to control an invasive species, or to be able to help with human health or human existence. So it's really the key to ecology is being able to analyze and use mathematical tools um, so that we can better predict what will happen when we change an environment. So the first key feature that we use to describe a population or to characterize a population, which should be obvious, is size. And so when we define the word population, we mean all of the individuals of a given species that live and reproduce in a particular place. So if we want to characterize what a population is, we can use size. And that would simply be the number of individuals of all ages alive at a particular time in a particular place. So let's use the example of krill. The population of krill in the cold waters around Antarctica is estimated to be at 800 trillion individuals, making them by mass the most abundant animals on Earth. And if we want to go a little bit further, we can then discuss the range and the density of the krill that are in a particular ecosystem. And when we talk about range, we mean geographic range. And we can define that as how widely a population is spread and the factors that determine its distribution, that population's distribution. So for example, the krill population that's made up of 800 trillion individuals ranges through an area of 19 million square kilometers around the continent of Antarctica. So we have population size, we have its range, the space where that population would be found on Earth, and the range of any species reflects the range of climates a population can tolerate and determines how many other species the population encounters. And that brings us to the next characteristic of populations, and that would be population density, which is defined as the population size divided by its range. If you think about density, that's how compact things are. And so if we want to determine a population density, we just need to determine how many individuals we have and the space where those individuals are kept or have access. And so in effect, population density tells us how crowded or dispersed the individuals are that make up the population. And for krill, 800 trillion animals are spread over 19 million square kilometers, which would give us a density of 42 million animals per square kilometer. One thing to be aware of is the fact that this way of characterizing population density as average density across the range of a population is often useful, but it can be somewhat misleading depending on how the organisms are distributed in a particular place. For example, the individuals within a population may be distributed randomly. That is, a new individual has an equal chance of occupying any position within the range or the space where it's living. And the location of one individual has no influence on where the next will occur. Sometimes, however, resources are distributed in patches within that space, or the chances of a new individual's survival are enhanced by the presence of other individuals. And in these circumstances, those populations may be clumped with individuals clustered together around those resources. In some other cases, one individual of a population might prevent another from settling nearby. For example, if all individuals depend on the same set of resources, then that might inhibit other organisms from being in that location. Or take, for example, predators searching for other members of the same species nearby. Under those conditions, individuals may be distributed more evenly than they would be predicted by chance. And so in that situation, ecologists would call those populations uniformly distributed populations. So in summary, we would define random populations as a population where a new individual has an equal chance of occupying any position within the range, and the location of one individual has no influence on where the next will occur. Clumped distribution would be defined as individuals clustered together in some small space. And this occurs when resources are distributed in patches within some sort of range, or the chances of a new individual surviving are enhanced by the presence of other individuals. And the third would be uniform distribution. And that's when individuals are distributed more nearly evenly than would be predicted by chance. And this occurs if all individuals depend on the same set of resources, or if predators, having located one prey individual, may search for other members of the same species nearby. And so the size of a population may increase, decrease, or it could even stay the same over time. And trends in population size are one of the key focuses of ecological research and the application that it has to conserving species that are in the world. A decrease in size, in the size of a population, for example, could tell us about the declining health in a local population, right? If the population is decreasing in size, then there might be a practical concern um, that we need to address. For example, if we're commercially harvesting fish, if all of a sudden that population has a very strong decrease in size, that is a key um, giveaway that something's wrong, or to understand which populations on the earth or which species on the earth are endangered. Those numbers would be able to allow us to determine that. In the 1990s, catastrophic declines in cod populations had an effect on the commercial cod fishing that went on in the Grand Banks of Newfoundland, and laws were put into place to ban the commercial fishing because the decline in the population size started to affect both the cod and cod fishers, and it led to changes in the ecosystems of the Grand Banks, affecting crab and oyster populations that eventually increased in size as a result because there were fewer cod that would prey on them. So many factors affect population size. An obvious process that contributes to a population increase would be birth. And another one could be immigration, for example. Individuals may arrive in a population from some other location. And then on the other hand, We'll have mortality or death and emigration, where that's individuals departing from a population. Those two would act to decrease the population size. So we have both the addition, the birth, and the immigration into a population that would increase its size, bolster it, and then mortality and emigration that would cause the population to decrease. And we have the influx and the influx of organisms into and out of that allow populations to either increase or decrease. Um, so individuals in many populations of butterflies and other animals most often live and die within the patch of habitat where they're born. So imagine we're trying to calculate the size of a population of lime swallowtails by counting every individual that existed in the Dominican Republic. That would take forever. So how do we know a population's size? We have to estimate. And like that of the lime swallowtail, the populations of most species are either too large or too widely dispersed for every individual to be counted. Instead, ecologists estimate population size by taking repeated samples of a population and from those samples estimating the total number of individuals. With an estimate of population size and range, we can then use that information to determine density. For sessile or sedentary organisms like milkweeds in a field, for example, or barnacles that are stuck to a rocky shoreline, biologists commonly lay a rope across an area of interest, and then they would count each individual that touches that rope. Or they could throw, for example, a hoop um, into some space and count the individuals inside it. Um, each such count would give one sample, and then they would take the number of samples and from that number estimate the number and distribution of individuals in the entire population. For example, suppose we had a meter squared hoop and we placed that in 3 different spots in the field. So we had 3 hoops, each 1 meter squared. And what we found out was that inside each of those hoops, we had a certain number of milkweeds, which is a plant. In one hoop, we had 4 individuals. In another hoop, we had 5. In another hoop, we had 3. We can multiply the average number of individuals, in this case, 4 individuals, per 1 meter squared hoop by the total square meters in the area the area of interest, in this case, the size of the entire field. And that would give us an estimate of the population size. And it's a relatively easy way of counting sedentary organisms along a straight line or within a grid area. But what we know is that many species move around. So the bigger question is, how do ecologists estimate population size for turtles in a pond or mice in a forest or butterflies? And the way that we do that can be done by a technique called mark and recapture, in which organisms are captured and then released on to different days. Butterflies, such as monarchs, are captured in an area. They're marked with some sort of sticker or a magic marker or something like that, and then they're released. And the number of butterflies that were marked that day is recorded. And then the next day, the researchers go out and they catch another sampling of butterflies in that same area. And the number of marked and unmarked butterflies that's recorded. The butterflies with marks are called recaptures because the day prior, those are the ones that the scientists had already captured and marked. And then in order to find population size, what we do is we take the total number of marked butterflies and unmarked butterflies caught on the second day, and we divide that by the number of recaptures. So we multiply this number by the number of butterflies marked on the first day, and we get this equation, n equals c divided by r times n, m. What does that mean? N in this case would be the population size, the number that we're looking for. C is the total number of marked and unmarked butterflies that were captured on day 2. R would be the number of marked butterflies that were captured on day 2. And then we would multiply that by the number of butterflies that were caught on day 1. And that doing the math would allow us to be able to predict the total number of organisms in that population. So let's say we capture 100 butterflies on the first day. We mark them, we release them. On the second day, we capture 120 butterflies, 30 of which are marked and the rest unmarked. So we would conclude that the population size is 120 divided by 30 times 100, which ends up being 400 butterflies in total in the population sampled. So these mark and capture techniques have been extremely useful in estimating population sizes of organisms from polar bears to disease victims, and we can even use them to be able to determine migration patterns and see where organisms are located on the earth and where they end up moving, or where they hibernate, where they spend most of their time, what they do most of the day, different things like that. So I have a video that I want to show you that discusses the migration patterns of monarch butterflies into Michoacan, Mexico. And it's a pretty cool video, so we'll watch that now.
The largest insect migration in the world ends each year in Michoacan, Mexico. Millions of monarch butterflies travel from across the US and Canada, some flying close to 3,000 miles to pass the cold months in the towering trees of this beautiful forest.
And so what we'll see here is an equation that looks somewhat complex, but I guarantee that it's easier than it looks. This equation allows us to be able to describe changes in a population size mathematically. And so we call it the population growth equation. And it is a way of determining how a population size changes over a particular time span. So we define a population size as a letter N, capital N, and the change in a population size over a given time interval as delta N, where the Greek letter delta, which is written as a triangle, means change in. So delta N is the number of individuals at a given time, we'll say time 2, minus the number of individuals at an earlier point in time, we'll say time 1, and we'll write that as delta N equals N2 minus N1. And if we expand upon that, we'll understand that the processes leading to a change in population size through time include the births of individuals, the deaths of individuals, the immigration into and the emigration out of. So we can determine the change or the population growth, either increase or decrease in a population. So we take the births and we add that to the number of immigrants entering the population, and from that number we subtract the deaths, and the number of emigrants leaving the population. And that will allow us to determine the change in population size from one point to another. So here's an example. Um, Ecologists often want to know not just whether population size is increasing or decreasing, but also the rate at which population size is changing. And so the change in population size in a given period of time can be calculated pretty easily. So if the question is, if a population starts with 80 individuals and grows to 120 after 2 years, what is the population increase per year? So we start with 80 individuals, it grows to 120, and it took 2 years to do that. So how would we calculate the rate? We would just take um, the change in population size, which is 40 individuals occurring in the span of 2 years, and we divide the two. So 40 divided by 2, which is 20. So we have a rate of 20 individuals per year. So we gain 20 individuals per year based on this population. Therefore, we are most interested in the proportional increase or decrease over time, especially the growth rate per individual, or the per capita growth rate. Per capita just means per person or per individual. So what we want to know is the proportional increase or decrease per individual for each individual in the population, the per capita growth rate. And here's an example. So if we want to figure out per capita growth rate, what we do is the equation that we have up here uses R. The per capita growth rate is symbolized by R, and it's calculated as a change in population size per unit of time divided by the number of individuals at the start. So R is equal to the change in population size over a certain amount of time divided by the population size at the beginning. And so in our example, R would equal 20 individuals per year divided by 80 individuals, because we had 80 individuals at the beginning, for a per capita rate of 0.2 per year, individuals per year. And it's important to realize that while rates are important in ecology, the data we usually have in hand actually consists of number of individuals that we've counted. And we don't know exactly how large a natural population really is, and so we have to rely on estimates. And we considered how we make such population size estimates about a couple of slides back. But for the rest of this discussion, we have to focus on the per capita increase or decrease in a population over time, and assume we know the population sizes for species of interest. If this per capita rate of increase were to continue, right? If it were to continue at a rate of 0.25 individuals per year that are coming from each individual in the population, how large would the population be after 10 years? So how would we calculate that? We use another equation. And this equation allows us to determine the per capita growth rate over time. So we can answer that question of how large the population would be after 10 years using the formula that's on the screen now. And solving this equation for our example shows us that in 10 years, the population size would increase to be 745. And how did we get that number? It would just be 1.25 raised to the power of, raised to the 10th power times 80, right? And that's written there. What about after 15 years? 15 years, what we would need to do is just plug those numbers into the equation, and where it says T, we put 15. So what was the population size at the beginning? 80. What is R? The per capita growth rate, that was 0.25. Plug all of those in, and we'd find out that after 15 years, the population would swell to 2,273. And this example is what we call exponential growth, the pattern of population increase that results when R is constant through time. A prominent feature of exponential growth is that the number of individuals added to the population in any time interval is proportional to the size of the population at the start of the interval. And so what I mean by that is while the per capita growth rate remains constant, the actual number of individuals added each time unit increases as the population grows. Why is that? That is because the individuals added will also eventually reproduce. And so through time, more and more individuals will contribute to the growing population. And on the next slide, I'll show you the characteristic shape of an exponential growth curve. And so let me show you that right now. And like I said, this pattern of population increase results when R is constant through time. And we call this exponential growth. If the per capita rate of increase we calculated in the previous example were to continue, how large would the prediction be after 10 years? We used that formula. We plugged in the numbers that we knew, right? The original population, which was 80, the R value, which was the per capita rate, which was 0.25, and T, which is the amount of time that we allow the growth to occur, which would be 10 years. And we were able to calculate what the population size would be at 10 years, which was 745, and then 15 years, and we could do it for 20 years and 30 years and so forth and so on, as long as R is constant. And so this is because the new individuals added also reproduce themselves. So each time individuals are added, they reproduce, and then their offspring reproduce, and their offspring reproduce, and so forth and so on, to make this exponential increase in number of individuals in that population. The per capita growth rate, R, is commonly called the intrinsic growth rate of a population. And we can define intrinsic growth rate as the maximum rate of growth when no environmental factors limit the population increase. This guy named Malthus recognized that exponential growth couldn't continue for long in nature, and the reason for that was because the number of individuals will eventually totally strip the resources available to support the population. And so when resources are gone, the individuals can't reproduce. And Charles Darwin used a lot of what Malthus wrote about and described when he talked about this exponential growth and the stripping of resources as population size increases to Darwin recognized that when the number of young produced at birth exceeds the number of adults that could be supported by available resources, what we'll see happen is that the individuals within the population will start to compete in order to obtain resources that are needed for growth and reproduction. And some of the competition could be within species, and some of it might be between species. When it's within a species, we call that intra-specific competition, and that results in natural selection. On the other hand, we might have individuals from different species also competing for resources, and then we call it inter-specific competition, and that can result in an increase or decrease in population size. What we see here are some examples of R values, high R values or low R values. And species that breed at a young age and produce many offspring typically have a very high intrinsic R value. So that would be fruit flies and E. coli and plants and that type of those types of organisms. And then on the other hand, we have species that take a long time to mature and have few offspring per year. They would typically have a low intrinsic R value. That would be like pandas or palm trees or humans or beech trees or cows. They would have lower R values. And so on this slide, we see what's called a logistic growth curve for a population. And this is similar to exponential growth, except something happens that caps the amount of or the number of organisms that can exist in a population. And that something could be resources, could be space. There are a lot of factors that could cap the number of individuals that can exist in a healthy environment. And so as a population grows, crowding, for example, commonly results in a decrease in the availability or quality of resources required for continuing growth. And as a consequence, the growth rate declines. On Hispaniola, for example, the lime swallowtails have probably reached the limit of available citrus groves, where the individual females have to search longer to find better suitable plants on which no other females have laid their eggs before. And with that delay and the pressure of eggs that are continuing to mature inside their bodies, the females end up laying too many eggs in one place or on poor quality food, and there isn't enough food for all the larvae to survive. And those factors uh, combine and they lead to fewer births per female in the lime swallowtail population that occurred immediately after they arrived in Hispaniola when resources weren't a limiting factor. But for many organisms, crowding affects the death rate as well as the birth rate. So, take for example a fish population that is increasing in a pond. There's less food available for each individual. Oxygen may be used up by the microbes that proliferate because of nutrients in the fish waste. In other words, we have birth and death rates themselves being affected by population density, which we defined earlier as the number of individuals per unit of area, or for aquatic species, like a volume of a habitat. But increasing density is not always detrimental to a population. For example, eggs of salmon or sturgeon are more likely to be fertilized in rivers or streams swarming with their species than when there are fewer reproducing adults. Generally, however, at some point, increasing population density causes the environment to deteriorate because of scarcity of resources. When the density of a growing population reaches this point, the rate of growth slows and eventually levels off as the death rate increases to equal or balance out the birth rate. And we call this maximum number of individuals a habitat can support the carrying capacity. And we use the letter K to represent carrying capacity. K represents that interplay between the requirements of individuals for growth and reproduction and the environmental resources such as food or space available to support those needs. And many different factors can keep a population below K, below its carrying capacity. Predation, parasitism, those are two examples. Another one would be so increasing numbers of, take for example, again, the lime swallowtails on Hispaniola. They've no doubt attracted predators and parasites that feed on them. Similarly, a high population density of fish in a pond increased the likelihood of infection by viruses, by bacteria, fungi, because those agents are transmitted from fish to fish. So the more fish we have, the more likely the bacteria, the more likely the fish, the more likely the viruses will bloom in those ecosystems as the density goes up. Predation and parasitism can reduce the population size below K, but they don't actually change K itself. On the flip side, many populations grow this way, and so logistic curves also apply to many other phenomena in biology, as well as I can think about it in chemistry and sociology and political science and economics. When we think about it, a logistic curve describes the rate that oxygen binds to a population of hemoglobin molecules, um, which we'll learn about later on in the year. Or similarly, the number of fertilized salmon eggs goes up with the amount of sperm deposited in a stream by males, but slows again as the percentage of unfertilized eggs available for the sperm to fertilize drops. As limits to growth due to these like density factors, these density dependent and density independent factors are imposed, what we see is the logistic growth model um, with populations where we have this huge exponential growth at the beginning, and then it starts um, to balance out, and we reach K, we reach that carrying capacity in the population. Um, what we see on this slide is a population. Um, we see the comparison of exponential growth with logistic growth, and we also see the carrying capacity. And I'm just going to read this quickly so you see it. But so a population growing at a constant rate without checks will increase exponentially. However, for most populations found in nature, as the population reaches its carrying capacity, the growth rate slows and eventually remains at or near zero. So when the growth rate is at zero, that just means the birth rate is equal to the death rate. The number of individuals coming into the population equal the number of individuals leaving the population. And that means we're at that carrying capacity where the population can be supported by the particular area, the resources in that particular area. So if we take that one step further, we can talk about fluctuation in size in a population and stability and what's going on at that carrying capacity. A term we use to describe that is population dynamics, which focuses on the interactions between biotic and abiotic factors that cause the variation in population size. A lot of long-term population studies have gone on, and they have sort of challenged the hypothesis that populations of large mammals are relatively stable over time because they're not always relatively stable. And what we can see is that weather or predator population can affect the size. I'm going to show you a video on the um, in two slides that discusses this example where we have a moose population on this island called Isle Royale that totally decreased. It collapsed during a super harsh winter, and what we saw was the wolf numbers peaked. But we'll see the reason for a moose population collapse and wolf numbers peak on this island in one slide. So this is a graph that shows us an example of density independent factors that could affect the population size in a particular range, so the density of a population in a particular area. At low density, food and other resources are not factors that limit growth. But what we see is that as density increases, they exert more and more influence on population growth, spurring competition for available resources. And similarly, at super high population densities, individuals may be more vulnerable to predation or to infection, so forth and so on. So factors such as resources and predation are called density dependent factors, whereas density independent factors influence population size without regard to the population's density. So those factors would include events such as a drought or a prolonged cold period, either of which could cause widespread mortality independent of the population's density. So let's look at the example on this graph here. This is called a population curve, or it's showing the population growth in an organism known as a song sparrow. And it's a function of the number of the growth is a function of the number of nesting territories established by the males each year. And here you can see that the number of territories generally increases for several years until an unusually dry spring occurs. And then at that point, there's a great decrease in the amount of vegetation available to support the insects on which the sparrows feed. And as a result, the number of territories then plummets, regardless of territory numbers the previous year. And so what happens is the number of territories declines following dry years. And then we see the song sparrow density declining consequently right after. So anytime you see the arrow on the blue line, that's showing a decrease in the sparrow density, which was caused by a decrease in the number of territories, which was caused by the decrease in precipitation in that environment that year. So these factors are affecting population, and they're not due to density. They're independent of the density of the population. This video that I'm about to show you discusses the wolves and the moose of Isle Royale, which is one of the longest predator-prey study that has been done in history. And what we see is that recently the wolf population on this isle has crashed, and it's leaving only 2 wolves, which are highly inbred, on that population. And the only way new wolves can access this Isle Royale is through an ice bridge across Lake Superior. But with climate change, this ice bridge that used to form 80% of the time now only forms 10% of the time. And the next ice bridge that occurs, so the next winter that we have, may be the last ice bridge that connects the main island, Isle Royale, to the general population of wolves. And that could essentially cause the population of wolves that are on this Isle Royale to go extinct. So a question I have for you to think about is, should our national parks do something about the effects of climate change if we're starting to see more and more of an effect of the climate, the change in the climate, causing extinctions and causing mass changes in the way the dynamics of a population. So we'll play that video now.
Isle Royale is a secluded 893 square mile island in the middle of Lake Superior. This park is one of the least visited national parks in the United States, but the most revisited because of its beauty and seclusion. It has no human inhabitants, but is home to many moose and wolves who significantly affect their surroundings. Isle Royale is the site of the longest-lasting predator versus prey study in the world. As late as 2009, there were 25 wolves on the island, but after the spring 2017 count, just 2 wolves remain. Because the remote island is isolated, the only way for new wolves to get to the park is over an ice bridge that can form over Lake Superior during cold winters. Climate change is making those bridges rare, says John Vucetich, a professor at Michigan Technological University.
Our understanding of ice bridges starts with going back to the 1960s. Ice bridges would have formed in about 8 out of every 10 years. More recently, they form in approximately 1 out of every 10 years. So from about an 80% chance of occurring in a year to about a 10% chance occurring in a year. Some projections would have the next ice bridge, whenever that occurs, could possibly be the last.
Because the wolf population is so low, moose numbers are increasing by about 22% each year. Vucetich studies predation ecology in Isle Royale. He is a leader in the Isle Royale Wolf Moose Project, which is in its 60th year.
That makes it the longest study of any predator-prey system in the world. And the centerpiece of that work has always been to count in the abundance of both wolves and moose. This winter, what we found out basically two main features: that the population was still just two wolves. We also understand that it's the same two wolves that had been there as the previous year. Those two wolves happen to be both brother and sister and father and daughter. They do seem to still be quite capable of killing moose, which means they can feed themselves. The other thing we learned about the wolf moose population this past winter is how many moose there are. There are approximately 1,600 moose on the island right now.
When a predator population crashes, the ripple effects of that crash are known as a trophic cascade. In 1994, Isle Royale became the first known terrestrial trophic cascade.
And so at the top trophic level are wolves, and what sometimes happens is that wolves affect the next trophic level, which would be the moose, by reducing their abundance. In reducing the abundance of moose, it causes the next trophic level below that, which is the vegetation, to do much better.
Mark Romansky, the division chief of national resources at Isle Royale, has worked on the island for almost 20 years.
So you can imagine without any wolves, moose are essentially allowed to do whatever they want, and that could mean browsing to unprecedented levels. Some of the larger sort of forest system-wide impacts is, with moose having impacts on browsing certain species, such as balsam fir. And some of the impacts include like changing the shape of these trees. Essentially, you have 1 meter tall trees that are 30, 40, 50 years old because they've been chronically browsed. And so we're not getting regeneration of new, of new balsam fir trees. And then that impacts also the type of forest community that you have over the long term.
The other really important creature that's affected would be beaver. Beavers will tend to be much more abundant when the wolves are not doing well, which is what's occurring right now. The beaver population is almost doubled in the last 4 or 5 years. And in doing so, they will make more beaver dams. And beavers in particular are what are sometimes referred to as architects of ecosystems. They really can change the overall structure of an ecosystem. They do this by making dams, and when they make a dam, they flood a river basin or a drainage, and then they start to cut the trees down that are in that area.
To bring back balance, the Park Service is looking at the reintroduction of wolves to the island. The possible reintroduction is leading to more questions about the National Park Service mission to protect the parks from human influence.
And if we're going to answer this question, whether we should bring wolves back to Isle Royale, we couldn't possibly answer it without first asking, what is the purpose of a protected area? What is the purpose of our national parks? And that's a pretty big and broad question, but in this particular case, what it hones in on is, what do we want to do with respect to climate change and our national parks? If climate change takes some things from national parks, are we okay with that? Will we just let climate change do that? Or if climate change takes things from our national parks and there's a chance to mitigate that or undo it, should we try to do that as well? That's a very big question. It's a question that goes much wider than just Isle Royale. And Isle Royale is going to be kind of an important, I guess for lack of a better word, test case for understanding whether we as an American people think it's good to try to mitigate some of these effects of climate change or not so much.
So the fate of the wolves of Isle Royale is in our hands. These are our national parks. This is Kim Leadholm signing off for the PBS NewsHour Student Reporting Labs from Isle Royale.
And just in summary, with regards to the video, what we saw is that the wolf, the moose, and the tree populations were all interconnected. It was the trees that provided food for the moose. The moose provided food for the wolves, and when the wolf population went down, the moose population went up, which resulted in slower tree growth. And so one thing affects another, and that affects another, and that affects another, and can have long-term ramifications in terms of the diversity of a population or the size of a population, which is due to climate change, which humans have caused. What we see here are what are called age-structured population growth diagrams, or age diagrams. And not all organisms in a population will contribute equally to growing that population because some organisms are more likely to reproduce than others. Therefore, it's useful to take into account individuals that have differing reproductive capacities, which more likely than not really depend on differences in age. So ecologists can set up this age structure diagram of a population to show the number of individuals within each age group of a population. And what we see is that a population in which most individuals are past their age of greatest reproduction, or a population mostly composed of individuals younger than this age, will not increase as rapidly as one dominated by individuals capable of the greatest reproductive output. And so estimating the number of individuals of differing ages then enables ecologists to be able to predict whether the size of the population will increase or it'll decrease. And what we see is that a growing population usually shows a pyramid-shaped age distribution like the one we see for India, with the youngest classes much more abundant than older classes, whereas a stable population shows a more distributed, more even distribution of age classes. And that's what we see in the example for France, which is a country that has a fairly stable population. In humans, ages in age structure reflect in part socioeconomic variation among countries. But differences in the age structure of other species, non-human species, could reveal threats from various sources. And the next slide that I show you will be an example of that. So the age structure of fish populations, for example, can be affected by the preferential capture of the largest fish, which is a reoccurring problem in commercial fisheries. Because what we see is the larger fish produce most of the eggs each year. And so when we capture them, this pattern eventually leads to the rapid decrease in the size of the population. And so here we see the effects of overfishing on the age structure of species of long-lived Pacific rockfish. Pacific rockfish typically live up to age 40 or more. And if we compare the ages of fish sampled in unfished waters with those in heavily fished waters, we see the effects of overfishing dramatically. In the unfished population, the curve shows many individuals close to and past the age of 10 years, when reproductive ability starts to decline. But in the fished populations, the curves show a sharp decline of individuals older than 6 years because mostly older and larger fish are the ones that are being caught because those older fish produce most of the eggs and young fish, and the annual reproductive effort of fished populations is severely affected by their removal. The age structure of a population could reflect death rates just as much as birth rates. And this thing called demography is the study of the size, the structure, the distribution of populations over time, and includes changes in response to birth rates, aging, migration, death. So let's look at the demography. Let's study the size, structure, and distribution of populations over time for the lime swallowtail butterfly that has been studied in Southeast Asia, where it's native. Because that investigation of how it grows and how it lives in Southeast Asia might help us better predict the population dynamics of the species in the coming years as it spreads through Hispaniola across the Americas. The factors that cause insect mortality commonly differ from one life stage to the next. So These butterflies, of course, start as eggs. They transition to young larvae. They develop and become older larvae. Then they go into the pupa stage, and then the pupa metamorphose into the adult lime swallowtail, which then produce eggs and so forth and so on. And so what we can see um, in this image that you're looking at are the effects that could potentially allow us to understand the physical and biological influences that that could change population growth. So what the image is showing us are the different dangers that at each stage of the lime swallowtail's life. The butterflies typically lay around 30 or more eggs, but only a fraction of those eggs survive because of parasitism or drying out, or because they fall from leaves, or predation by ants or other insects. And the larvae have um, many defenses against predators, such as camouflage, or they have chemical toxins, um, but they still fall prey to wasps, as well as lizards and some other animals. So the faster that they develop into adults, the more likely they are to escape discovery by their enemies. The pupa, which are large and immobile, those are perhaps the most vulnerable. That's the most vulnerable stage for the lime swallowtail life cycle, because they're often the most highly parasitized uh, of the life cycle of the organism's life cycle by a particular kind of wasp that comes in and attacks them. Um, so what we can see, if I show you on this slide, what's a life table and a survivorship, are data on the duration of each stage in the lime swallowtail life cycle and the numbers of um, organisms that are killed at each stage or that die at each stage. So let's start in the egg stage, and we'll call all of the eggs that are present at the beginning of the study one group. Um, we'll call that a cohort, which is basically just a group of individuals a group of individuals born at a given time. And we can trace their survival as they age and as they metamorphose. The information about how many individuals of a cohort are alive at different points in time can be summarized in what we call a life table, from which an ecology can identify patterns of survival in a population. The proportion of individuals from the initial cohort that survived to each successive stage of the life cycle is called survivorship. So what we see is the graph shown here is what we call a survivorship curve through time that shows for these butterflies a declining trend, starting with the total number of eggs at the beginning. And we can see in the life table and the survivorship curve for this example that survival is lowest among the eggs. 68% survive to the next stage, and pupa only 45% to the next stage, which of course the pupa can't move to escape any harm. So if we divide the number lost at each stage by the number of days that the organism spends in that particular stage, we can see that the egg stage loses 16% per day. The pupa stage loses 6 per day, but the larva stage only loses 0.6 per day. There is therefore strong selective pressure to decrease the length of each stage, especially those stages that are most vulnerable to mortality, such as the pupa stage. Nevertheless, if we remember that each adult female can lay 30 or more eggs, only a few eggs need to survive to the adult egg laying stage to ensure the growth of the population. And in warmer climates like the Caribbean, the butterflies may reproduce 8 or 9 times in a year. Most of their eggs will not survive to become adults. If they did, the resources that are available would be depleted, and we would see a balancing off of the population as it reaches the carrying capacity for that environment. Populations differ in the ages at which they experience the most mortality. In some species, many offspring are produced, but high mortality early in life means that few survive to reproductive age. In other species, mortality is highest in older individuals, while in some others, mortality occurs with equal frequency at all stages. What we can see from the example shown here are the three principal types of survivorship curves, type 1, 2, and 3 from left to right, which correspond to the pattern of mortality. Whether most mortality occurs late in life, is even throughout life, or is concentrated early in life, is what we see in each of those three curves. Human populations in affluent countries experience most mortality later in life. And so typically it shows a type 1 survivorship curve, which is the one on the far left. Other large animals, such as elephants, or you can think of whales, show similar survivorship curves. And then in contrast, we might have birds and other small mammals commonly experience consistent levels of mortality throughout life, which could be graphed as a declining straight line. That would be the curve that's shown in the center. These animals, for which the probability of death doesn't really change through the life cycle, they would exhibit what's called a type 2 survivorship curve. And then the last one, the one on the far right, could be organisms such as turtle grass or algae. That would be a high mortality at the earliest stages in life cycles, resulting in sharply declining survivorship curve that eventually levels off. This pattern is what we call type 3 survivorship curve, and it's typically of small herbaceous plants, small animals like mice, or could be insects. The lime swallowtail, for example, is type 3, or other organisms that grow fast and reproduce early and then eventually just die off. So some species produce large number of offspring, but provide few resources for that offspring support. And in that situation, we call those species R strategists, lowercase r dash strategists. On the other hand, we have species that produce relatively few young, and they invest considerable resources in the support of their young. In that case, we call them K strategists. And how do these concepts relate to the K and the R that we talked about before in population growth? And the answer to that is that, in general, species that live in unpredictable environments produce many young. When the environment is full of predators watching for vulnerable prey, such as the gulls that watch for young turtles on a beach, or when we have resources distributed in patches and abundant for only a short period of time, the chances of a young organism's survival to adulthood are low. Laying an egg, then, is likely a lottery ticket. There's a limited chance of success. It's more advantageous to buy many tickets cheaply, that is, to have many offspring and make little parental investment in any of them, than to pay a lot for one. So, thus, salmon and other fish produce many thousands of young, each with an extremely small chance of survival. And as we saw earlier, organisms like insects and plants that display type 3 survivorship curves also experience high mortality in the first stages of life, because their reproductive success depends on the small chance that a few of their young will survive all the hazards they encounter. The parents produce many offspring and invest relatively little, if any of them. So we refer to species like most fish as R strategists because they reproduce at rates approaching R, the intrinsic rate of growth. And then on the other hand, species living where resources are predictable, they often produce a lower number of offspring and invest their reproductive resources in just a few, often larger, they're better cared for offspring. And those offspring are therefore better protected from predators. They're able to compete with others when they're young, and they're more likely to survive bad weather. Large animals, think of eagles, humans, bears, they produce only 1, 2, 3 young each uh, each birthing, and each of which is much more likely to survive to reproductive age than the offspring of small animals like mice, which have many predators and are easily killed by cold weather. So we would consider eagles, bears, humans K strategists because their population densities commonly not lie near carrying capacity, which is K, when resources become scarce. Um, these two strategies reflect an evolutionary trade-off. Think of it this way: a reproductive individual has access to a finite amount of resources and can um, invest either in many expensive young or few well provisioned, well protected offspring. And depending on the combination of physiology and environment, natural selection in a given set of circumstances may favor one strategy over the other. However, R and K strategists represent the opposite ends of a continuum in reproductive patterns. Many species lie between the two extremes. Um, and how an organism makes use of the resources that are available can also affect population growth. Each individual must devote some of its available food and energy intake to growth, some to the maintenance of cells and tissues, and some to reproduction. In nature, individual organisms strike a balance in how they allocate resources to basic needs of growth, maintenance, reproduction, a balance that has evolved to minimize waste while retaining the flexibility to use resources in different ways depending on the circumstances of the environment. The typical pattern of resource investment in each stage of a given species lifetime is what we call life history. Um, the amount of energy available for reproduction varies over an individual's lifetime. And the degree of control that an individual has over resource allocation also varies from one species to another. So to go on with a little bit of the term life history or the typical pattern of resource investment in each stage of a given species lifetime, I have an example that I want to talk about. And this example talks about the different populations of fish that live in streams. We have lots of guppies, and that's the image that you see on the top, living in a stream with many small waterfalls that serve as somewhat natural barriers that keep them in place in isolated populations. But some guppy populations face different predation pressures. Some populations live with these fish that are called killifish, and it's a relatively small predator that eats small guppies. Other populations live with a different type of fish called a cichlid, a cichlid, which are larger and they tend to eat more mature guppies. So the question I have for you is, how does predation affect the life history traits in guppies? How do the organisms that are living in that environment affect the traits of the guppies? And so what we have to realize is that many species are subject to different types of predation at different stages in their life cycle. In some cases, the predation pressure can influence life history traits. And here's a study that was done. So this is a long-term study by evolutionary biologists where they wanted to illustrate the effect of predation on life history traits. And what they did was they studied populations of guppies, which are, like I said, small fishes that are native to shallow freshwater streams in the mountains. And the study was done in one particular region where many small waterfalls prevent guppies in one part of a stream from moving to another. And as a result of these natural barriers, each stream holds several population of guppies that have very little gene flow between them, meaning there's no immigration into or out of those individual populations. The waterfalls also keep guppy predators from moving from one part of the stream to another. The main guppy predators, like I was saying, are the killifish and the cichlids, and they differ in size to they differ in size and prey preferences. The relatively small killifish preys on the immature guppies, and they ignore the larger adult guppies. Whereas the cichlids, they like the bigger fish. They tend to pursue the mature guppies, and then they ignore the smaller ones. So some parts of the stream hold one type of predator, but not the other. And what we see is different guppy populations face different predation pressures. And the researchers discovered that guppies in regions with cichlids grow faster and are smaller at maturity than guppies in regions with killifish. And guppies in population hunted by cichlids also reproduce earlier, having more offspring at a time and breed more frequently. Were these differences in life history traits genetic, or did some environmental variation cause them? And that is the question that these researchers had. So, in order to find that out, what they did was they collected guppies from both the cichlid dominated and the killifish dominated streams, and they reared those groups in separate aquariums under identical predator free conditions. And 2 generations later, the groups continued to show the differences observed in natural populations. And the researchers concluded that those differences between guppies preyed on by different predators have a genetic basis. They hypothesized that the predators act as selective agents on guppy life history patterns, and they made a prediction. And they said that if life history traits evolve in response to predation, then these traits will change when a population is exposed to a new predator that favors different prey traits. So, in order to test that, they found a stream region above a waterfall that had killifish, but no guppies or cichlids. And to this region, they introduced the guppies from a site below the waterfall where there were cichlids but no killifish. And at the experimental site, the guppies that had previously lived only with cichlids, which eat the large guppies, were now exposed to killifish, which eat the smaller ones. The control site was a downstream region below the waterfall where relatives of the transplanted guppies still coexisted with the cichlids. And when the researchers revisited the stream over the course of 11 years and 36 generations of guppies, what they saw was um, they recorded some data that showed that the guppies at the upstream experimental site were evolving. And exposure to a previously unfamiliar predator caused life changes in the guppy's rate of growth. It changed the age at first reproduction and some other life history traits of the guppies. And then, in contrast, the guppies at the control site showed no changes. They concluded that life history traits in guppies can evolve rapidly in response to selected pressures exerted by predation. Another example is the hare lynx population, and we call this cycling. And when we analyze population cycles, we are able to get a pretty productive way of understanding how intraspecific that means in a particular species, and inter, that means between different species. We get a good way of being able to understand how those two density dependent factors interact. And in this example, we will see how a classic case of population cycling, which involves 2 species, the snowshoe hare and the lynx in northern Canada, has changed over time. What we'll see on the x-axis on the graph, plotting a 50-year interval, and the y-axis showing changes in hare population density on the left in a log scale, and changes in lynx density on the right. Data for the hares are plotted in tan. Data for the lynx are plotted in blue. And there are 3 key points that I want you to know about. The scale and the y-axis are different. There are many more hares than lynx. The y-axis for hares is logarithmic to compress the values so they can be compared more easily to changes in the lynx population. The hare and the lynx population cycle every 10 years on average. And then the third key point is that changes in lynx density lag behind changes in hare density by about 2 years. So the snowshoe hares are herbivores and they subsist on leaves and stems that grow close to the ground, whereas lynx are predators and they subsist mainly on the snowshoe hares. And to explain this cycle, the biologists um, posed two hypotheses based on density dependent factors. It's the first hypothesis known as the bottom up hypothesis, and that's that idea that when their populations reach high density, hares use up all their food and starve. In response, then the lynx also starve. And there is another hypothesis that's called the top down hypothesis, and that's that lynx population reach high density in response to increases in hare density. At high density, the lynx have access to so many more hares that the prey population eventually crashes. In other words, either hares control lynx population size or lynx control hare population size. This cycle could be controlled by intra-specific competition for food or inter-specific interactions, or it could be an interaction of both. And so there was a study that was done in order to test this and see exactly what was going on, and that is right here. I already described the process, so I'm just going to skip over this slide, but you can pause it and read the information if you'd like. And this last video that we're about to watch is by E.O. Wilson, who is a biologist who helped formulate a theory that describes how species diversity can differ depending on an island where the species is located, where that population is located. And islands represent extremes in habitat patchiness for colonizing species. In other words, organisms that live on a mainland must successfully cross a barrier of water or something else in order to establish a new population on an island. And a lot of times, ecologists expand the use of the term island to include any habitat patch that is surrounded by a substantial expanse of inhospitable environment. While this use of the term island certainly includes land surrounded by water, the way we normally think about it, but it can also apply to a body of water surrounded by land, or it could be a part of a forest surrounded by grasslands, or a mountaintop surrounded by forests at lower elevation. So one big question that they had was, where do colonists of these habitat islands come from, and what determines how many succeed in establishing a new population? And what they came up with was this idea known as the theory of island biogeography in 1967. And the theory of island biogeography states that the number of species that can occupy a habitat island depends on two big factors. The first is the size of the island where they are inhabiting. And because of the size, what we would say is larger islands receive more colonists than smaller ones, and larger islands support more species than smaller ones, so the extinction rate is lower. And for those reasons, the equilibrium number for species for larger islands is greater than for smaller islands. And the second key factor for determining the number of species that can occupy an island habitat would be the distance that that island is from a source of colonists. The more distant islands have lower rates of colonization, moving equilibrium species diversity towards lower values, right? The species diversity would be lower the farther the island is from a source of colonists.
In the late 1950s and and early 60s, as I was trying to figure out the organization of ant society, I was also thinking about bigger natural systems, ecosystems. Not many people thought like that back then. How, I wondered, did different species fit together to form an ecosystem? A natural catastrophe gave me the key. On August the 27th, 1883, one of the greatest volcanic eruptions ever recorded laid waste to the Indonesian island of Krakatoa. It was, they say, the loudest explosion ever heard on Earth. Over 36,000 people were killed. Krakatoa was one of the worst natural disasters of modern times, but exactly because it was so destructive, it was an unexpected gift to later generations of biologists. Life on the island had been completely wiped out. That tragic event made Krakatoa the perfect laboratory in which to find out how species colonized and recreated an ecosystem. And biologists quickly, even in the 1800s, saw the value of studying what kind of species would come back and settle Krakatoa and what kind of relationships they might have. Dutch naturalists visited the island repeatedly in the years after the eruption. They carefully chronicled the returning species. It was a grand experiment, and we have been able to use the data to this day. Those data reveal the fascinating fact. Almost exactly the same number of bird species repopulated the island as had inhabited it before. The species were different, but their number was the same. This seemed to show that ecosystems naturally find their own equilibrium. It made me think there might be a simple mathematical formula expressing this balance. But to confirm my intuition, we needed a more controlled experiment. So I decided to create my own mini Krakatoa in the Florida Keys. We went down to Florida Bay, where there are thousands of little mangrove islands, little dots on the map. We got an exterminator from Miami. We covered a number of islands different distances from the other islands so they could be fumigated by the same technique used to fumigate warehouses in order to eliminate all of the creatures on this little island. Please don't think of me as destroyer of biodiversity. They were just one out of thousands. We could then carefully record the rate at which insect species recolonized the little island and their number. The results strikingly confirmed our hypothesis. That all ended up in a book by Robert MacArthur and me called The Theory of Island Biogeography.
Ed Wilson and MacArthur were the first people to actually quantify and create a theory of how communities behave on islands, how islands become populated by organisms, and what happens next. And they were the first ones to actually specify those rules and build equations that allow us to predict what will happen with organisms on islands.
We figured out how to predict the number of species that will arrive and live and form an ecosystem on an island. But the theory didn't just apply to islands. I wanted it to apply to all ecosystems and the cause that's closest to my heart, conservation.
Island biogeography has a tremendous application in conservation because as we modify the environment around us and leave behind little patches here and there of the natural habitat, we are actually creating functional islands of those natural habitats.
Later, after MacArthur and I had published a theory of island biogeography, it became a centerpiece in ecology and also in conservation biology. You know, the planning, the study, the understanding of ecosystems. As I've come to understand even more about ecosystems, I've devoted more and more of my energy to the cause of biodiversity. The natural world has always been my greatest love. If a biologist can't fight for it, who can?
So with regard to island biogeography, a useful calculation is that a tenfold increase in island size would result in a doubling of species diversity. And I think phrasing it another way in terms of conservation makes that concept a little bit easier. So if I say there's a 90% reduction in habitat area, that means that we will lose 50% of the species diversity in that area. And that's a huge amount of species diversity. And this relationship between island area and species diversity has so many applications in conservation biology. For example, determining how large a protected area must be to support species, or how close reserves have to be to one another to ensure the movement of individuals between them, and so forth and so on. So this theory of island biogeography has been widely used for conservation biology, but it does have some limitations. For example, the theory treats all potential colonists as equally able to colonize a new island, and that's not necessarily true. Let's take, for example, the animals, flying animals like bats and birds. They would more frequently arrive on oceanic islands than non-flying animals like rodents, lizards, and frogs. And then on the other hand, non-flying animals that colonize successfully more often evolve into multiple species on islands. And if these new closely related species can't themselves colonize another area, they will be found nowhere else in the world, and we call them endemic species. So the example that you see on the screen right now is an example of island biogeography for the anole lizards, which is a great example because the Anole lizards um, inhabited an island close, mainland close to the island of Hispaniola, and there were only a few number of events, maybe 5 separate events, that caused some Anole lizards to make their way onto this island. And as a result of those few events, we have um, an explosion of diversity, dozens of species of Anole lizards present on that island today. And they're all marked with huge evolutionary diversity that happened after the colonization. So pre-colonization, the anoles, in terms of diversity, looked pretty similar, and then after colonization, there was an explosion because of the bottleneck um, of traits and different niches where the anoles could live and um, get resources. So what we see is that the anoles have evolved about 6 different feeding strategies, and each feeding strategy has um, corresponding morphological traits that the organism has, that the species has. All 6 types can coexist in one place because they hunt insects in very different ways, which means that the anoles are highly diverse. Take, for example, the crown giant anole. The crown giant anole, it has morphological traits that allows it to get its resources from the crown or the canopy of trees in an environment. Or take, for example, the grass bush anole. They are highly adapted to get their resources, eat from the grass and the bush. Or you take the trunk anole, and the trunk anole can get its resources from the trunk of trees, eat insects that are found in that area. But they're highly adapted, and they have lots of different characteristics that allow them to survive and reproduce best in very different um, areas of the island. And that um, is really due to this theory of island biogeography, and um, this example illustrates that pretty well. Um, and so that finishes our conversation about population ecology. I'm super tired, so that means I am logging off. But if I can answer any questions, or if I can help you in any way, please let me know, and I will be sure to reach out.